This time, we're going to continue our discussion of cellular respiration by looking at stage 2, the citric acid cycle. Remember, during glycolysis, glucose was broken down into the chemical pyruvate, which then entered into the link reaction so long as oxygen was available and was converted into acetyl CoA. So, in the second stage, the acetyl CoA is now going to act as your starting substrate. So the initial reaction is the acetyl of the acetyl CoA will combine with another chemical, oxaloacetate, and that's what makes citrate or citric acid. Remember, we are talking about a cycle, so eventually the citrate will be broken back down into oxaloacetate in order to repeat the reaction. So what we'll see is a series of enzyme-controlled reactions where the citrate or citric acid is gradually converted back into the oxaloacetate, which is then free to combine with more acetyl-CoA, make more citrate, and repeat the cycle. And as we do these reactions, we produce carbon dioxide as a waste product and ATP, our energy molecule, our desired product. All of this is taking place within the matrix of the mitochondria. We noted in the glycolysis video that NAD was collecting hydrogen molecules to be passed on to the third and final stage of respiration. And we see that again here in the citric acid cycle. So the dehydrogenase enzyme, it removes hydrogen from different chemicals and passes it to the coenzyme NAD. So that is how we form NADH, as NAD acts as a hydrogen carrier, picks up that hydrogen from the dehydrogenase, carries it to the third stage of respiration. So dehydrogenase enzymes are found in glycolysis and in the citric acid cycle to remove those hydrogens, passes to NAD, and that's how we get our NADH. So that's everything we need to know about stage 2, the citric acid cycle. So let's try some more past paper questions. So here the list shows some of the substances produced during the respiration of glucose in the presence of oxygen. 1. An acetyl group, 2. Pyruvate, 3. Citrate, and 4. ATP. But which of the following sequences shows the order in which these substances are produced? Pause the video and hit play when you're ready to reveal the answer. Okay. So to answer this question, remember both glycolysis and the citric acid cycle will have taken place. So the correct answer is A. So during glycolysis, we make ATP during the energy payoff phase, and that leaves us with the end product pyruvate. The pyruvate then gets converted into acetyl during the link reaction, and the acetyl eventually gets converted to acetyl CoA, combines with the oxaloacetate, and that's how we get the citrate during the citric acid cycle. Let's try a different one. So here you're asked to state the function of the dehydrogenase enzymes. Okay. So again, pause the video, come up with your response, and hit play to reveal the answer. So let's take a look at the marking scheme for this one. So the role of dehydrogenase enzymes are to remove or to release hydrogen. So do not accept collects hydrogen. That's not the enzyme that's doing the collection, that's the NAD. The dehydrogenase enzyme is what removes the hydrogen. Another way you could have worded this is to say it passes the hydrogen to the coenzyme NAD. So hydrogen removes dehydrogenase removes hydrogen from the chemicals and passes it to the NAD molecule. You could also have used some shorthands here. They accepted H or H plus to represent the hydrogen or the hydrogen ions, or if you were specifically talking about the electrons, you could have used E with this little negative symbol. So remember, abbreviations can often be used. So hopefully you've got a better understanding now of the first two stages of cellular respiration. And next time we'll cover the third and final stage, the electron transport chain.